These are some of the important terms we use in this lab. Genetic engineering, the direct manipulation of gene for practical purposes. In this case, we used a genetic engineering technique called transformation, a change in genotype and phenotype due to the assimilation of external DNA by a cell. And the gene we tried to transform was green fluorescent protein. It's a protein that absorbs blue light and produces green light. And this is from jellyfish. This is our hypothesis. In control plate with E. coli only will live. Plate with E. coli and amplicillin will die. And in transformation plate with E. coli, amplicillin, and PFG DNA will have some growth but will not glow. Plate with E. coli, amplicillin, PFG DNA, and IPTG will have some growth and will glow.
These are brief steps to our experiments. In two test tubes, one that says negative and positive. The negative means negative DNA, which is a control tube without PFG plasmid DNA. Positive is our transformation tube containing PFG plasmid DNA. First, we added ice cold calcium chloride to each test tube, 250 microliters each, and then we scrub some colonies out from LB sources and transferred to each of these tubes. And then we added 10 microliter of p-fluorogreen DNA, PFG, to the test tube labeled positive. And then we put these both tubes into the ice for 10 minutes, and then we heat shock them by putting it into 42 degrees Celsius water bath for 90 seconds. And then we put it back to the ice buckets for 2 minutes, added 200 microliter of LB L'Oreal recovery broth to each test tube and gently mixed. And then we added back to them in 30-70 degrees Celsius water bath as a recovery period. And we transfer each of these test tubes in these plates according to the instructions.
These are the 4 plates that we produced from these steps. These 2 are control groups and these are transformation groups. The one on the top of the control group is just the E. coli cells. And this one, it has the E. coli cells and ampicillin. And for the positive groups, which is transformation groups, the top one has DNA, ampicillin, but no IPTG. But this one has DNA, ampicillin, and IPTG. These 4 plates were placed in incubator, which is 37 degrees Celsius for overnight incubations.
After letting the bacteria incubate for 24 hours, we will see the results. These 2 trays do not have the plasmids. The one on the left that just has LV, it is just regular bacteria and food, so they grew a lot as seen in the picture. The one on the right, they don't have plasmids and they have the LV and the antibacterial on it, ampicillin. And the one on the right has no growth because they have no plasmids, so they have no resistance to the antibiotics, so they all die. In the second set of trays, they did receive the plasmid because both trays have the ampicillin on them, and once they received the plasmid, they had a resistance to the antibiotic. And in this picture you can see each plate had a small amount of growth. There were about 5 or 4 colonies on each individual one. On this one we put it under the ultraviolet light. It has the plasmids, it has the LV and ampicillin. There are a few colonies, but they did not glow. there's a little smudge on the outside, but the bacteria colonies themselves were not glowing. On this one, this tray also has the IPTG, which unactivated the repressor gene, so it allowed the bacteria to produce the green fluorescent protein, so they did glow as seen on this picture under UV light. In this lab, we observed transformation take place in these bacteria cells. Looking at each of the plates that did not get the plasmid did not receive the ampicillin-resistant gene, so it cannot survive on the plates with ampicillin present. The plate without ampicillin grew well since it had no antibiotic limit to it. The plates with plasmids grew only on the plates marked plus and with ampicillin. Only the few colonies that grew had the plasmids because if they would not survive the antibiotic. We also calculated the transformation efficiency from this experiment. In the tray that had the plasmids but not the IPTG, they had 5 transformants in the entire tray. Up to the calculation, they have 2 times 10 to the 2 transformants per nanogram, and the IPTG one, they have 4 transformants on the tray in total, and they had 1.6 times 10 to the 2 transformants per nanogram. This shows how the actual transformation curve is actually really low, and it is not a very efficient way to have transformation. Our results supported our hypothesis. Our hypothesis included that the plain plate with E. coli cells will only live, and it was true, they lived, and the plate with ampicillin with the bacteria cells, they died. And the hypotheses for the transformation plates, we believe that the plate with ampicillin with E. coli cells and the PFG DNA will have growth but not green, and those were the results we saw. And the last hypothesis was the plate with the ampicillin with E. coli and PFG and IPTG will have some colonies and will also go green. And this was true, and this is another picture of someone else's results, but it shows the same colonies and the difference if the repressor gene is suppressed or not suppressed. The purpose of the control plates was to make sure the experiment was working correctly and that the bacteria grew, and that they also would not grow if the presence of ampicillin was present. There were possible areas for error in this experiment involving contamination or possibly mixing up the mixtures. We also, another possibility is mixing up the bacteria and also mixing up the plates, some which had the antibiotic on it or not. And this picture just shows some examples how the green fluorescent protein is being used in science. And here are some websites that I used to get the pictures for this experiment. Thank you.